All right, hello everyone. Today we will be covering chapter 21. It's not really a chapter on a specific system. We'll just be covering like a special topic. It's over water, electrolyte, and acid base balance. The first part of the chapter is kind of relatively easy, it's more anatomy based. But the second part of the chapter is more difficult. A lot of students kind of struggle with it. It's more acid-base balance. So we'll be talking about pH and the hydrogen ion concentration, um, and buffer systems and how we kind of maintain homeostasis with our bloodstream and how hydrogen, different ways hydrogen ions maintain homeostasis in our body. So most students find the second part of the chapter a little bit confusing. So just remember that as we go through the lecture. Here are some roots, prefixes, and suffixes that you will need to know throughout the chapter. Again, most of you are familiar with some of these. Edema is swelling. Anemia is usually a condition associated with the blood. Here it says hypoproteinemia is a low protein blood count. Hyperproteinemia would be high protein concentration in the blood. Hypokalemia, low potassium. Hyperkalemia, high potassium. Intra, within. Osis. We'll talk about acidosis and alkalosis. All right. Okay, so water and electrolytes. We've mentioned this several times now that water and electrolytes are interdependent in the body. Just the word interdependent just means that electrolytes or ions are dissolved in water. And that usually a change in one causes a change in the other. And when we talk about the electrolytes, the important ones that we're talking about in the body are going to be sodium, potassium, calcium, hydrogen, hydroxide, chloride, magnesium. And everything that we talk about, the body has to maintain homeostasis. We don't want too much sodium. We don't want too little hydrogen. We don't want too much magnesium. And we have to have this balance. We talked in the kidney chapter that this amount of water loss, that 180 milliliter, or the 180 liters that get filtered out each day, and obviously we don't pee out 180 liters of water, and about the majority of that gets taken back in the body so that we don't excrete too much water. And same thing goes with electrolytes. A lot of electrolytes get filtered out. And then we do the whole reabsorption and renal tubular secretion and reabsorption so that our urine is concentrated so that we keep what we need to keep our body functioning. All right, so with our body fluids, they're not going to be uniformly distributed. We do have different compartments of body fluids, and they do have different compositions. We'll talk about the different compositions here in the next slide, but we do know that water and electrolytes can move between those two compartments just to keep everything stabilized and homeostatic. And we do know that there are differences between males and females, and the females have about 52% water by weight, and males have about 65% water by weight. Unfortunately, females have about have a little bit more adipose tissue than males do. Males have a little bit more muscle tissue. And adipose tissue actually has a lower water content. And muscle tissue has a higher water content. So that's why males have a higher water content by weight, because they typically have higher amount of muscle tissue. Um, all right. So when we look at our different compartments, we can separate them into intracellular fluid compartment and extracellular fluid compartment. Remember, we talked about the ICF and the ECF. And in the average male, we're only going to talk about the male here. We're not going to break it up into male versus female, but typically we have about 40 liters of water in a typical male, and we can break that up into the ICF and the ECF. And you can see that in the intracellular fluid, it's about 63%, and the extracellular fluid, it's about 37%. So if we do the ratio, we're about it's about 2 to 1. Inside of our cells, it's approximately 2 to 1 inside of our cells to outside of our cells. So intracellular fluid is all of the water and electrolytes inside of our cell membrane, inside the actual cell itself. Extracellular fluid, 30%. Outside of our cells, that's in our interstitial fluid, our interstitial spaces, the tissue spaces. Remember that graph that I like to keep going back to, where we push 20 liters out per day and we pull back 17 liters, and those 3 liters go into the lymphatic system. So that's outside of the cell. So that's where your interstitial fluid is. In your blood plasma, the fluid, your plasma is not inside of our cell. That 3 liters that's left over, that goes into your lymphatic system, that's outside of our cell. We haven't really talked about transcellular fluid yet. Transcellular fluid, again, it's not inside of a cell, but it's kind of separated from, it's not free floating like in plasma or in interstitial fluid, but it's still separated by epithelial membranes or epithelial layers. So think of like your aqueous humor and your vitreous humor that's in your eyeball. It really doesn't migrate outside of your eyeball. Your cerebral spinal fluid doesn't really migrate to your big toe. Synovial fluid that's in like your knee joint, it really doesn't migrate outside of your knee joint. So it's kind of like isolated to a specific area. So those are examples of transcellular fluids. They're just kind of localized to one specific compartment or one specific area. But since they are not inside of a cell, they are still considered part of your extracellular fluid component. All right, so here's just another picture graph of breakdown of where we could see water in different compartments. So 63% of your fluid is going to be intracellular, 37% is extracellular fluid, and you can see most of the fluid is going to be in your interstitial space. Some of it's going to be in your plasma. Some of it's going to be in your lymph, and the remaining is going to be in your transcellular fluid. Remember, transcellular fluid, that's your cerebral spinal fluid, synovial fluid, your aqueous humor, vitreous humor, serous fluid. Remember, serous fluid is that, you have your parietal layer and your visceral layer, and you have that thin layer of serous fluid that kind of decreases friction. Remember, parietal and visceral layers, one lines the organ, your visceral layer lines the organ, your parietal layer lines the cavity, and your serous fluid lay is that watery fluid between those two layers to decrease friction. Okay, and so with your body fluid composition, remember that we don't just have water, but we have electrolytes that are dissolved in water. And we can see that typically your extracellular fluid is very similar in composition. And when we look at the cell versus the extracellular fluid, you can see that there's a higher concentration of, I'll change my pen color here. So you can see that there's a higher concentration of sodium outside of the cell. There's a higher concentration of chloride outside of the cell. There's a higher concentration of calcium outside of the cell. And there's a higher concentration of bicarbonate outside of the cell compared to inside of the cell. And I guess I should say that that blue circle thingy is a cell. And when we look at the cell itself, we have a higher concentration of potassium inside the cell compared to outside. We have a higher concentration of phosphorus inside than outside. And we have a higher concentration of magnesium and sulfate inside the cell compared to outside of the cell. So if we were, let's say that, we have nothing else going on, if we're talking about simple diffusion, if we're talking about simple diffusion, we would say that sodium would move from the outside to the inside. We would say that potassium would move from the inside of the cell to the outside. We would say that calcium moves from the outside to the inside because of the area of higher concentration to the area of lower concentration. All right, and remember that blood plasma contains more proteins than interstitial fluid or lymph only because the large proteins don't escape those blood vessels. Remember, the blood vessels don't allow those proteins to escape. They don't have those large holes that allow proteins to escape those blood vessels. All right, and if you remember from that slide that I like to reference a lot, we have factors that allow movement of fluid to go in and out of blood vessels. And those two factors are hydrostatic pressure and osmotic pressure. Remember, hydrostatic pressure is the pressure that allows water to move back and forth, or that allows for it's all going to be movement of water. But hydrostatic pressure is the force of water, and osmotic pressure, and we can call it colloid osmotic pressure, plasma colloid osmotic pressure, protein pressure. So we have a lot of different names for osmotic pressure, but osmotic pressure is the pressure that's exerted by the proteins themselves. And even though glucose isn't a protein, because glucose typically doesn't escape the blood vessels, because remember, glucose typically doesn't spill off into the urine. Glucose does provide or does contribute to the osmotic pressure, even though glucose is not a protein. Osmotic pressure is that protein pressure that is one of the driving forces to help pull water back into the blood vessel. So when we look at movement of fluid between the compartments, between the intracellular compartment and the extracellular compartment, what we see is that the hydrostatic pressure remains relatively equal. So unlike the graph that I like to represent, or that I like to look at with the arterial end, where we have that 20 liters going this way, and the 17 liters going this way, and then we have that 3 liters that go off into the lymphatic system. That's not what we're talking about here. What we're talking about here is just the movement of fluid between the intracellular compartment and the extracellular compartment. So we're talking about movement of fluid between inside of the cell and outside of the cell. And what we see is that that hydrostatic pressure is pretty much the same between those compartments. So if we look at the water pressure or that hydrostatic pressure between the difference between what's on the inside versus the outside of the cell, that hydrostatic pressure is essentially the same. So we're not going to see a big difference in that hydrostatic pressure between the inside of the cell and the outside of the cell. So the driving force for water movement or fluid movement between the intracellular compartment and the extracellular compartment is going to be osmotic pressure. So most of the fluid results from changes in osmotic pressure when we're talking about fluid movement between compartments, the intracellular compartment and the extracellular compartment. And remember, osmotic pressure is going to be proteins and glucose. And remember, it says impermeate solutes. And remember, impermeate just means that it can't cross that cell membrane. That's what impermeate means. So just some key takeaway from the slides. One, you're going to need to know that your concentrations of what is greater on the inside of the cell versus the outside of the cell. You're going to need to know those. And you're going to need to know that, one, the hydrostatic pressure is going to be relatively equal on the inside of the cell versus the outside of the cell. And the driving force is going to be osmotic pressure, which is going to be made up of your proteins and glucose. All right, and here's just a picture representation of what you can see with your concentrations. So we said in this last slide that sodium is going to have a higher concentration outside of the cell than inside. So here, extracellular fluid is blue and intracellular is orange, yellow, orange, I guess. So here you can see that that sodium has a higher concentration outside than inside. Potassium has a higher concentration inside than outside. Calcium, even though there's not much of a big change, calcium has a higher concentration outside. Magnesium has a higher concentration inside. Chloride has a higher concentration outside. Bicarb has a higher concentration outside. Phosphorus and sodium. So sodium has sulfate has a very relatively small concentration difference. And then here is just on the right-hand side, you can see just the picture representation of kind of what we were talking about with the hydrostatic pressure and the osmotic pressure. And here is the hydrostatic pressure forces fluid into the capillaries, and the interstitial fluid is now equilibrium. All right, so with water balance, obviously, we need to maintain homeostasis, and theoretically, our water intake should equal our water output. If we drink 3 liters, theoretically we should pee out 3 liters, or we should put out 3 liters. Now, we do have some sort of metabolic production of water. Remember, when we do cellular respiration, we produce energy, carbon dioxide, and water. Those are when we do glycolysis, citric acid cycle, we produce ATP, energy, carbon dioxide, and water. So we do have to take that metabolic production into account, and everything is needed for homeostasis. But, we should not see a big adjustment. And of course, we have to take into account, what is this water output? Not only is water output what we pee out, but, if we go running, we might sweat. When we breathe out, we actually breathe out a little bit of water. So we have to take that into account as well. And it's also not only our water intake is what we drink, but we take in water in foods that we eat as well. So water intake, we do have thirst centers in our brain. We'll talk about that a little bit later. And our water output is primarily controlled by the kidneys. Not obviously, if we're sweating, that's not going to be really controlled by our kidneys. But we also have this thing called a diuretic. We talked about ADH, that antidiuretic hormone. But anyway, a diuretic just means to promote urine production. We talked last chapter about caffeine, and citric acid is also a diuretic, and we can give medications to help us get rid of some fluid. If you're like in a congestive heart failure, if you have hypertension, you might give Lasix or Bumex or something like that to kind of get rid of some urine as well. All right, so for water intake, and of course, this is going to vary among individuals, and especially if you're a patient on dialysis, this is not going to apply for you because you don't want to take in a lot of fluids if you're on dialysis, because that can throw you into congestive heart failure rather quickly. But for an average adult who's healthy, you're going to take in, and again, if you're a runner, if you're doing a lot of exercise, you're going to probably bypass this 2,500 milliliters, but you're going to take in about 2,500 milliliters of water per day. Now, 60% of that is going to be from drinking fluids, and that's about 1,500 milliliters. 30% is going to be from moist foods, or about 750 milliliters. So think about if you eat an apple, or if you eat a peach or an orange, they're very juicy. So that's going to be some water intake there. We also have 10%, I told you, as a byproduct of cellular respiration or oxidative metabolism of nutrients. We call that metabolic water. So that's what I talked about up here, metabolic production. It's called metabolic water right here. So remember, we produce carbon dioxide, water, and ATP. So we actually produce about 250 milliliters of that per day. And if we add up the 1,500, 750, and 250, we should get 2,500 milliliters. And again, this is just an average. Some people will drink more, some people will drink less. All right, so for water output, so if we take in 250, 2,500, we should be able to get rid of 2,500. Now, that's going to be about 60% in the form of urine. 6% is going to be in feces. 6% lost in sweat. We call that sensible perspiration, because even though we might not be doing anything, we're still going to sweat. But 28% is going to be lost through evaporation from the skin, called insensible perspiration, or from the lungs during breathing. And then, of course, we know that, if it's hot and humid outside, if we have a different activity level, that's going to adjust or alter how much we're going to sweat out. And we do know that it's going to, if we adjust our water intake, it's going to adjust our output as well. And this is just kind of like a picture representation of just what we talked about. All right. All right, so water intake. So obviously, you're going to drink water when you are thirsty. And we have these thirst, we have this thirst center in our brain. But it's actually because we have a change in our volume, basically our blood volume, or the volume in our extracellular fluid compartment. So we have a change in our blood volume, or what we could say is we could have a change in our osmotic pressure. Remember, the hydrostatic pressure really doesn't change at all. And that osmotic pressure is what is the driving force that we learned kind of way back up here in this slide. So the osmotic pressure is going to kind of be the driving force for fluid movement. So our thirst sensation is derived from either a change in volume, like blood volume, or in osmotic pressure changes from that extracellular fluid compartment. And that osmotic pressure, remember, is due to changes of impermeant solutes, proteins, and glucose that cannot cross the cell membrane. And remember, everything such as, a lot of your negative feedback loops are going to go through your hypothalamus. So we have these osmoreceptors that are going to go through your hypothalamus that detect changes in your osmotic pressure of your body fluids. So we have receptors that are in our cells that are going to look at what is the osmotic pressure of the cell, and it's going to throw those values up into our brain, and they're going to get processed by the hypothalamus. And the hypothalamus is going to have these osmoreceptors and say, hey, we're in range, everything's good. Oh, nope, we're out of whack. We need to drink. Or, oh, nope, we're too high, or we're good, or no, we need to quit drinking water. We have too much water on board. So in our hypothalamus is basically where our thirst receptors lie. And even if we lose only 1% of our body water, that's going to be what triggers or stimulates our thirst sensation or our thirst center. And it looks like I don't know how to spell thirst. Thirst. So only losing 1% of your body water will trigger or stimulate those thirst centers or those osmoreceptors in your hypothalamus. So your osmoreceptors in your hypothalamus are going to detect an increase in your osmotic pressure. We also have stretch receptors in our blood vessels that are going to detect that water loss. So remember, we have changes in blood volume, or we have changes in osmotic pressure that will kind of trigger our thirst center. So here we can have either our osmoreceptors kind of being activated, or we can have our stretch receptors in our blood vessels. So if we, let's say we accidentally cut ourselves kind of rather badly, and we lose 1% of our blood loss, we're going to trigger our thirst centers. Or if we donate blood, typically when you donate blood, you're actually going to donate about 10% of your blood volume. So if you donate 10% of your blood volume, you actually lost 10% of your body water, 10% of your blood volume. So you're going to trigger your thirst sensation. But also a decrease in your blood pressure will activate that renin-angiotensin system that we talked about as I believe in the last chapter, the kidney chapter. But angiotensin 2, remember we talked about angiotensin, but that angiotensin 2 will trigger your thirst center as well. That thirst, those thirst receptors in your hypothalamus. Angiotensin 2 will also trigger you to drink fluids. But just note that once we drink fluids and our stomach actually distends a little bit, that's going to inhibit our thirst center. So let's say that you have like a gastric band. Let's say that you had gastric bypass surgery, or you had that gastric band placed to kind of decrease the volume or the amount that your stomach can hold, and you drink, just a tiny cup of water, so the gastric band reduces the volume that the stomach can hold, and you drink a little bit of water and it stretches the stomach with that lower volume. So therefore less water intake is needed to satisfy thirst with a gastric band. So it's going to also inhibit that thirst center. So with the angiotensin and that renin-angiotensin system, that's going to stimulate the release of aldosterone and ADH, if you remember from the kidney chapter, or the urinary system. And remember that aldosterone will lead to the retention or the reabsorption or the conservation of sodium. And remember that where sodium goes, water is going to follow. So aldosterone leads to the conservation of sodium and the excretion or peeing out or tubular secretion, whatever you want to call it, of potassium. So we're going to keep sodium and we're going to get rid of potassium. And also ADH will lead to the retention of water. All right, regulation of water intake. So we've already said 1% of water. Once we lose 1% of water, we're going to have an increase in the osmotic pressure. So if we have, remember that we have, let's say, a cup of water with a bunch of proteins in it. And if we remove some of that water, we didn't remove any of the proteins, we just removed some of the water. Therefore, the amount of proteins is going to remain the same, so the concentration of proteins is going to increase, even though we didn't increase the number of proteins. So because we increase the concentration of proteins by removing the water, we're going to have an increased concentration of proteins or an increased osmotic pressure. And that increased pressure is going to stimulate those osmoreceptors in the hypothalamus. It's going to cause us to feel thirsty. We drink water. And that distension of the stomach is going to inhibit the thirst center. And that water is going to get absorbed to the stomach, and then we're going to go back homeostasis. All right, regulation of urine output. So remember that that the distal tubules and collecting duct, so that those last two portions of the nephron, the distal tubules and collecting duct, are the effectors of mechanism that control the amount of water excreted in the urine. And remember that that's where ADH works, that antidiuretic hormone. ADH works in the distal convoluted tubule and the collecting duct. And remember, ADH, anti-diuretic hormone or anti-P hormone. So if we need to hold on water, ADH is going to work at the distal convoluted tubule and the collecting duct. So we have these osmoreceptor ADH mechanism again in the hypothalamus because it's a negative feedback loop. It's going to regulate the concentration of urine. So we have in dehydration, we're going to have water loss. Those osmoreceptors are going to lose water because we're dehydrated. They're going to shrink up a little bit. That's going to stimulate ADH secretion. So now we're going to hold on to water. Remember, ADH will lead to the retention of water in the distal tubules and the collecting duct. And then now we're going to have water. Now we drink excess, or after drinking excess water, those osmoreceptors are going to swell. We're going to inhibit our ADH, causing water to stay in the distal convoluted tubule and stay in the collecting duct. We're not going to reabsorb water. We're going to keep it in the distal convoluted tubule. We're going to keep it in the collecting duct. And it's going to leave the body as urine. And again, remember, we have, we can lose water through sweat, through breathing, through urine, and through feces as well. But water loss through urine is going to constitute the greatest percentage of water loss. Now, one thing that I will say here is that alcohol and ADH. Alcohol actually inhibits ADH release. So remember, ADH, anti-P hormone. Alcohol inhibits ADH. So therefore, alcohol will increase the volume of urine that is produced. So alcohol inhibits ADH and will increase the amount of urine that is produced. All right, so here you can see what happens during dehydration and excess water intake. We kind of just covered this in the last slide, but one thing that, and I'll let you read this one on your own. We're not gonna go through it because we kind of just went through it on the last slide. One thing that you'll have to remember is that if you remember from chapter 13, the endocrine system, the posterior pituitary is what releases ADH. Remember, the posterior pituitary does ADH and oxytocin. Let's see, what else? If you drink too much water, osmoreceptors are going to gain water. They're going to swell up, and ADH is inhibited. All right. All right, so just some water balance disorders. Obviously, dehydration. It's when we have a deficiency of water in which output exceeds intake. So that could be prolonged vomiting or prolonged diarrhea. It's where our extracellular fluid becomes concentrated. It could be where our thermoregulation mechanisms fail. We have lack of water for sweating, could result in hypothermia, and our wastes can accumulate. Typically, we're not going to see long-lasting effects of dehydration. Just drink some water, you're okay. We'll see long-lasting effects of dehydration, usually in the hot summer. Nursing home patients, in the older population, you might see news reports about about dehydration, hypotonic hydration, water intoxication. We've actually seen some of this kind of pop up in the news not in more like in the past year. Water intoxication, more of like I've seen two. Not necessarily water intoxication. Well, one was water intoxication, where the guy drank 3 gallons of water in like 30 minutes and ended up dying. And I think this was after like a hot dog eating contest. But another one wasn't water intoxication, but more of it's hypotonic hydration, where the mom, I think it was a mom, and she was at a beach, and she was really dehydrated, and she drank like 4 bottles of water really quick, and they weren't the pH of the water was off a little bit, because it was like a frou-frou bottle of water, and so she drank these bottles of water real quick to try and get, because she was thirsty, and she ended up, I think she ended up dying because of it. Basically, there is an imbalance between the water and electrolytes. And if you remember way back when, we had this example of what happens when you place a solution, or when you place your red blood cells in an isotonic fluid, and a hypertonic fluid, and in a hypotonic fluid. And this is what happened: is she ended up drinking this water and placed her body into a hypotonic situation. And basically the electrolytes were too diluted to maintain this fluid distribution. And basically the water kind of rushed into her red blood cells, which caused her red blood cells to swell up and burst. So anyway, and then edema. We've talked about edema before. Usually that's because of decreased plasma proteins. Remember that colloid osmotic pressure that pulls that 17 liters of fluid back into the venule end. If we have low protein concentration, we can't pull that 17 liters back in, so we leave some fluid in that interstitial space, and that can lead to edema. Or it could be because we have obstructed lymphatic vessels. Remember that the 20, the 17, and we have that 3 left over that goes into the lymphatic vessels. If we have an obstruction that in those lymphatic vessels, that fluid can't go into the lymphatic space, into the lymphatic vessels. Inflammatory process. One thing that's not on here is Addison's disease. Addison's disease is basically an aldosterone deficiency. And if you remember aldosterone is retention of sodium and excretion of potassium. So with aldosterone, we don't have that retention of sodium, so we're going to lose extracellular sodium, and where sodium goes, water's going to follow. So we're losing sodium, so we're going to lose water as well, and that's just what Addison's disease is. All right, so we talked about edema. So here's some factors associated with edema that we talked about. Low plasma protein concentration, obstruction of lymph vessels, increased venous pressure, inflammation. We're not going to spend too much time here. All right, electrolyte imbalances. Basically, we need to keep, our electrolytes in balance. If you go for a run, don't just drink water, throw in some Gatorade as well. Kidneys are typically pretty good about keeping our electrolytes in line. We need to maintain homeostasis, but you need to do your job into replacing those electrolytes. We already talked about the electrolytes that are most important in our body. You can see those there. We get most of our electrolytes from foods, water, beverages, but we can produce some of those from metabolism of foods that we eat. If we do have an insufficiency, we might develop some hunger or thirst. Like if we don't have enough salt in our diet, we might crave salt. All right, electrolyte output. Again, kidneys are good with maintaining the electrolyte balance. We know that when we sweat or perspire, electrolytes get discharged through our skin. Some electrolytes are lost through our feces. All right, regulation of electrolyte output. Most of your positively charged ions or your cation ions are going to be vital for nerve impulse conduction. We talked about calcium and how that's good for nerve impulse conduction. Remember that calcium is needed for the vesicles to fuse to the cell membrane to dump out the neurotransmitter. Calcium's good for in those T-tubules for skeletal muscle contraction. We have that sodium-potassium pump. All right, potassium is for nerve, cardiac muscle cells. All right, sodium accounts for almost 90% of the positively charged ions in extracellular fluid. If we go back up to this one, if you look at how much sodium is in the extracellular fluid, if we just look at the positively charged ions, sodium, potassium, calcium, and magnesium, if we look at the concentration of sodium, potassium, calcium, magnesium of the extracellular fluid, sodium is going to constitute about 90% of what is in the extracellular fluid. Aldosterone, remember, aldosterone is going to remember that aldosterone retains or conserves sodium and excretes potassium. So aldosterone is going to be stimulated by low sodium levels. So therefore, we're going to see tubular reabsorption or conservation of sodium. And it's also stimulated by high potassium levels, because remember, we conserve sodium and we excrete potassium. So aldosterone can be stimulated by two things: low sodium or high potassium. And then calcium levels stimulate secretion of PTH, or parathyroid hormone, or parathormone, which is going to cause an increase in the plasma calcium levels. We'll actually see this kind of later on. We'll talk about this this calcium and parathyroid imbalance a little bit. Let's see, in a few slides. But all right, so electrolyte balance. This is just a slide that shows that we can take in electrolytes and they go out through perspiration, feces, and urine. Here you can see how just kind of what happens with our regulation of electrolytes with potassium, aldosterone, what happens with our sodium, potassium, parathyroid hormone. All right, so sodium and potassium imbalances. So hyponatremia is low blood sodium concentration caused by prolonged sweating. So obviously, when you sweat, not that I go recommend licking your sweat, but we all know that sweat tastes kind of salty, sodium. So caused by prolonged sweating, vomiting, drinking too much water. Because remember that sodium has a high concentration in the extracellular fluid, so its effects are hypotonic extracellular fluid, hypernatremia, which is high blood sodium concentration. That can be caused by excess water loss, fever, diabetes insipidus, or insufficient ADH secretion. Remember, antidiuretic hormone. So antidiuretic hormone insufficient, so we're going to pee out too much water. So if we pee out too much water, we're going to be left with too much sodium in our blood. Now, if we have too much sodium in our blood, we can see that in our central nervous system. Too much sodium could eventually lead to confusion, stupor, or coma. Hypokalemia, low blood calcium, and hyperkalemia, high blood calcium. Those actually both can cause life-threatening cardiac abnormalities. But one thing that I wanted to talk about, hypercalcemia, it's not on this list, but hypercalcemia, we talked about this in the bone chapter. Hypercalcemia, remember that thyroid senses that the calcium is going to be too high, and the thyroid is going to release calcitonin, and those osteocytes, or the osteoblasts, are going to tell the osteocytes to deposit calcium in bones. Or the osteocytes are going to tell the osteoblasts to deposit calcium in bones, and blood calcium is going to go down. And then we have hypocalcemia, and this is where the parathyroid senses that the blood calcium is too low, and the parathyroid is going to release PTH, and the osteocytes tell the osteoclasts to release calcium from the bones, and the blood calcium levels go up. With hypocalcemia, low blood calcium levels, all you need to do is just eat calcium, eat some calcium salts, but remember that you're going to need to take vitamin D supplements because it's going to optimize that intestinal calcium and phosphorus absorption. So if you're on calcium supplements, you need to be on vitamin D supplements to help absorb that calcium in the small intestine. Now, with hypercalcemia, we have this thing called kind of like chronic hypercalcemia, and I mentioned this like on that slide that we'll talk about kind of this a little bit later on. So with chronic hypercalcemia, we have the high calcium levels in the blood. It can also be seen with like overactive parathyroid glands. Normally, your parathyroid senses if the calcium is too low, and the parathyroid releases PTH, and that your osteoclasts tell your osteocytes tell your osteoclasts to release calcium from bones, and your blood calcium levels go up. And this process is going to continue and continue. If we have overactive parathyroid glands, you might see stunted growth due to the constant osteoclastic stimulation. All right, so if you do have chronic hypocalcemia, if you have insufficient dietary calcium, your bone matrix would remain soft from all of the calcium that's coming out of your bones. All right. Let's see. All right. So this is just kind of what I was talking about with the mom who drank that hypertonic or the hypotonic hydration, and that was on the beach. And even though it was like frou-frou water, but here we have normal hydration in this first beaker. The second beaker is hyponatremia, and here is hypernatremia. So what the mom did on the beach is that she drank a bunch of water that contained that put her in a hypotonic hydration state. So the electrolyte balance became kind of out of whack, and all of this water kind of rushed. Her fluid and electrolyte balance was off, so she actually had kind of like water overload. And so this water kind of just went into her red blood cells to try and dilute the ions in her red blood cells, and caused her red blood cells to burst. All right, so acids are substances that ionize in water and release hydrogen ions, or release an H+. So phosphoric acid, sulfuric acid. Usually they're designated. You can usually readily identify them. They start with like an H in their chemical formula. H2SO4. H3PO4, so something like that. Bases, on the other hand, are substances that typically release ions, usually an OH minus, that can combine with those hydrogen ions. And bases usually are going to lower the free concentration of hydrogen ions in body fluids. So bases are going to kind of neutralize the acid in your body fluids. So acid-base balance involves the regulation or maintenance of hydrogen concentration, of the hydrogen ion concentration in body fluids. And it's important to maintain homeostasis because slight changes in hydrogen ion concentrations can alter the rates of a lot of your metabolic processes, a lot of your enzyme-controlled metabolic reactions, hormone actions, ion shifts. So if you look at like a lot of protein structures, a lot of proteins only work within a certain pH range. And if your pH falls outside of that range, the proteins, or if your blood falls outside of a certain pH range, the proteins aren't going to be functional. So remember that pH, the pH is going to determine whether or not a solution is acidic or basic. And we can say that basic also means alkaline. And the more acidic solution, the lower its pH. The more basic the solution, the more alkaline solution, the higher its pH. And so we know that a pH of 7 is on the pH scale runs from 0 to 14, and a pH of 7 is neutral. And so anything less than 7 would be acidic, and a pH of anything greater than 7 would be basic or alkaline. And when we talk about the blood, typically the pH of blood is going to be 7.35 to 7.45. So we could say that the normal pH of our internal environment, or the normal pH of our body, is 7.35 to 7.45. And if we look at, and different areas of our body have different pHs. If you look at the pH of our gastric content, or gastric acid, it's going to be a lot lower than 7.35 to 7.45, because we know gastric acid is, pH of like 1.5 to pH of 3, somewhere around there. And when we do cellular respiration or any type of metabolic reaction, we're going to produce hydrogen ions. And here you can see how some of the processes that can produce hydrogen ion. So in the top row, these are going to be the processes. So in the top row, these are processes. So there's going to be a test question about, which process is going to produce hydrogen ions. And there's going to be some distractors, like carbonic acid, lactic acid, anaerobic respiration, aerobic. So make sure you choose one of these processes and not carbonic acid or lactic acid. So during aerobic respiration, such as like the citric acid cycle, remember glucose is broken down in the presence of oxygen to produce carbon dioxide, water, and energy. Carbon dioxide then combines with water in the blood to form carbonic acid, and carbonic acid is H2CO3. And this reaction is facilitated by that enzyme called carbonic anhydrase, and we talked about that already in a previous chapter. Carbonic anhydrase, or carbonic acid, then dissociates into bicarbonate, H2CO3 anions, and hydrogen ions, or H+. And this reaction helps regulates the pH of the blood and maintains the body's acid-base balance. So remember that aerobic respiration of glucose releases hydrogen ions into body fluids. During anaerobic respiration, like glycolysis, glucose is broken down in those 2 pyruvate molecules, which goes into the citric acid cycle. We talked in chapter 9, I believe, about oxygen debt and how this normal process is interrupted. Remember the electron transport chain and those NADHs that we talked about? So in lactic acid fermentation, NADH transfers its electrons directly to pyruvate, generating lactic acid, or generating lactate as its byproduct. Lactate is just the deprotonated form of lactic acid. And from here, lactic acid dissociates into lactate ions and hydrogen ions when dissolved in water. Incomplete oxidation of fatty acids in the liver produces ketone bodies, which can be used for energy. This process is called ketogenesis and occurs primarily in the mitochondria of liver cells. Liver produces ketone bodies during periods of, like, let's say you restrict your calories, fasting, low food intake, carbohydrate restrictive diet, starvation, prolonged intense exercise, alcoholism, untreated type 1 diabetes, mellitus. Ketone bodies are water soluble molecules that are transported into tissues outside the liver where they're converted into acetyl-CoA and oxidized for energy. And the brain can use ketone bodies for energy metabolism when glucose is scarce, such as times during stress. We're not really going to talk about this oxidation of sulfur-containing amino acids or hydrolysis of phosphoproteins and nucleic acids. All right, so acids, remember things that, remember acids are anything that ionize in water and release hydrogen ions. So we can have strong acids and weak acids. Strong acids typically ionize more completely and they release more hydrogen ions. An example would be your gastric acid. Gastric acid, like I said, is a pH of 1.5, like to 3, somewhere around there. Weak acids, like carbonic acid, are going to ionize less completely, and they're going to release fewer hydrogen ions. And I actually don't need the ions there because the H plus is inherent, meaning ions. So carbonic acid is an example of a weak acid, and carbonic acid can dissociate to produce carbon dioxide when combined with water. Bases, on the other hand, strong bases ionize more completely and they release more OHs or other negatively charged ions that can combine with the H pluses. Like a strong base would be sodium hydroxide, and weak bases are any bases that ionize less completely, and they can release fewer hydroxyl groups or other negatively charged ions that combine with, I guess I should write that, combine with with hydrogen ions. An example would be sodium bicarbonate, and that bicarbonate can act as the base. All right, and remember I said that the pH likes to be between that 7.35 to 7.45, and either if we shift outside of that range, that could endanger our internal environment. A lot of our normal metabolic reactions could be altered. And typically when we talk about acid-base balance, if we have too much acid or too little acid, too much base, too little base, we can eliminate hydrogen ions, or we can get back into homeostasis in one of 3 ways. We have this chemical buffer system. We have a renal excretion of hydrogen ions, and we have a respiratory excretion of carbon dioxide. This chemical buffer system, we're going to call that our first line of defense in shifting that pH, only because it's very rapid. Our chemical buffer system can rapidly correct pH changes. All right, so buffers, we have a chemical buffer system, and buffers are just are in all fluids, and they're chemicals that combine with excess acids or excess bases, and they're substances that stabilize the solution when an acid or base is added, and they minimize pH changes in body fluids. And what they do, I forgot to advance your slide. There we go. They minimize pH changes in body fluids. What they do is they're going to convert strong acids to weak acids. They convert strong bases to weak bases. Remember that strong acids are going to, and I'll abbreviate strong acids as SAs, and strong bases as SBs. So strong acids are going to ionize to release a lot of hydrogen ions. And in the presence of a buffer, we're going to convert those strong acids to weak acids so that we can minimize the release of hydrogen ions so that we can keep our pH in normal range. And then on the flip side, we can convert strong bases to weak bases. So remember, strong bases release a lot of hydroxyl groups so that they can so that the strong hydroxyl groups can bind with a lot of hydrogen ions to form water. So let's say we want to keep our hydrogen ions. Let's say we're too alkaline and we need to be more acidic. We could use our pH buffer system to get rid of our strong bases and convert them to weak bases to kind of keep our hydrogen ions kind of in our body to lower our pH, to get it back to an acidic side. So we can use buffers to convert strong acids to weak acids to get rid of our hydrogen ion concentration, or we could use our buffers to convert strong bases to weak bases. We have this thing called a bicarbonate buffer system that's found in both the intracellular and extracellular fluid. Bicarbonate is going to act as a weak base that is going to convert a strong acid to a weak acid. And it can also convert a strong base to a weak base. And here's our formula there. And we saw this formula back in the respiratory chapter when we looked at how carbon dioxide gets transported in the bloodstream. And we see that this is a double arrow because it can go to the right or to the left depending on what our body needs at any given time. So if we have too much acid in our body, we're going to go this way. We're going to get rid of hydrogen ions. Hydrogen ions are going to get taken up and we're going to form carbonic acid. If our conditions are too basic, we're going to go to the left and we're going to break down carbonic acid into bicarbonate, and we're going to release hydrogen ions to make our pH more acidic. So a lot of our equations that happen in our body, or a lot of our metabolic reactions that happen in our body, are the double arrowed reactions so that they can go to the right or to the left depending on what our body needs are at the time. We also have a phosphate buffer system that's also found in both the intracellular and extracellular fluids. But these are going to be found more in the renal tubules and in the urine. And so here, where's my cursor? All right, there it is. So in the presence of a weak base, this HPO4 is going to act as a weak base when we have excess hydrogen to form H2PO4. And then H2PO4 is going to act as a weak acid, and it's going to release hydrogen ions to turn the condition more acidic. So again, we have that double-headed arrow, so that if we have acidic conditions, we can go to the right and create more H2PO4. And if we have more basic conditions, we can go to the left and create more hydrogen ions. So your phosphate buffer system is going to work more in the kidney area and with the renal tubules. All right, we also have a protein buffer system. When we're talking about plasma proteins like albumin or hemoglobin, certain cellular proteins, remember proteins get broken down into amino acids. And if you remember the structure of the amino acids, we have all right, for the structure of amino acids, remember we have the so we have a C, H, then we have this R group that makes the amino acid different. We have a COOH, and then we have an NH2 group. So that's kind of like the general structure of an amino acid. So what we can do is we can buffer, depending on if we're too acidic or too alkaline or too basic, we can either release or take on another, we can either release a hydrogen ion or take on another hydrogen. So here we could actually get rid of this hydrogen to release that hydrogen, or if we need to take on a hydrogen, if we have a free-floating hydrogen, we could actually put that hydrogen right there. And that would kind of help with our buffer system by altering the pH of the blood to adjust our pH. All right, so here are just the three different buffer systems, the bicarbonate, phosphate, and protein, and kind of what they do, and we've already kind of already mentioned those. All right, so the second one is the when we go back up to, we can come back up to this time. So it's this slide. So we talked about the chemical buffer system. The chemical buffer system was the bicarbonate buffer system, the phosphate buffer system, and the protein system. So now we're going to talk about the respiratory excretion of carbon dioxide. So the respiratory center, we know is located, excuse me, it's located in the brainstem, and it can regulate hydrogen ion concentration body fluids by controlling the rate and depth of breathing. Remember that our carbon dioxide level is directly related to our hydrogen ion concentration. We learned that in the respiratory system or the respiratory chapter. So that if our carbon dioxide goes up, our hydrogen ion goes up, our hydrogen ion concentration goes up, therefore our pH goes down. And if our carbon dioxide level goes down, our hydrogen ion concentration goes down, and our pH goes up. So what happens is that if we hold our breath, our carbon dioxide is going to rise because we still have the cellular metabolism going on. So we hold our breath, our carbon dioxide is going to increase, which means our hydrogen ion concentration is going to increase, which means our pH is going to go down until we blow off some of that carbon dioxide. So increased production of carbon dioxide by the body cell is going to lead to the formation of carbonic acid. And we learn from that buffer system that that carbonic acid is going to dissociate into the hydrogen ion and the bicarb. And we have that equation here. We have this equation here that we learned in the respiratory system. So in response to increased acidity, excuse me, in response to increased acidity, or in response to a decrease in plasma pH, the respiratory center can increase the rate and depth of breathing. So if we have an increased acidity, that means we have a low pH. That means we have a high hydrogen content. That means we have a high carbon dioxide. We can increase our rate and depth of breathing so we can blow out some of our carbon dioxide, which means if we blow off some of our carbon dioxide, we're also going to blow off we're going to get rid of some hydrogen ions as well. We're going to alter that kind of equation. So by changing our breathing rate, the partial pressure of carbon dioxide of the blood is changed, leading to a change in blood pH. So remember that carbon dioxide and pH go in opposite directions. So an increase in carbon dioxide production means a decrease in our pH, or carbon dioxide, hydrogen ion concentration, pH. Carbon dioxide, hydrogen ion concentration, pH. All right. So remember that example that we talked about in the respiratory chapter about, I'm going to hold my breath until I die. Well, you really can't do that because you're eventually you might pass out, but eventually you're going to trigger your brainstem to take a breath. And remember that carbon dioxide is our drive to breathe, not oxygen. And if you remember from the respiratory system that, we have that oxygen hemoglobin dissociation curve and the partial pressure of oxygen, and we actually have enough oxygen in our bloodstream or bound to hemoglobin to last us a while, which is why our new CPR algorithm kind of focuses more on chest compressions as opposed to breathing, because we need to get rid of that carbon dioxide. So basically, we need to focus more on this carbon dioxide as our drive to breathe. All right. Renal excretion of hydrogen ions. So this is our third one. So remember that from the kidney chapter, we learned that hydrogen ions are actively secreted throughout the entire nephron. And the hydrogen ions are excreted in the urine. And we didn't learn that. We really didn't learn, what gets secreted where and what gets reabsorbed where. But hydrogen secretion is linked to tubular reabsorption of bicarb. And that's going to go back to kind of like this equation right here. Because we want to kind of keep everything in balance, homeostasis. But we have this renal buffering system to prevent everything from becoming too acidic or too basic. Because this is a kidney system, this renal buffering system, renal means kidney, we can call this a physiological buffer system. And then we can also, when we go back up to this slide, we didn't talk about these last two, the oxidation of sulfur-containing amino acids and the nucleic acid, the hydrolysis of phosphoproteins. We really didn't talk about those, but that's proteins can produce excess when we break those down. But we're really not going to talk about those. All right, so when we do take in proteins, remember proteins get broken down into amino acids. We're just going to kind of run through those a little bit. Amino acids can increase our hydrogen ion concentration, which eventually is going to get excreted into the urine, which maintains our pH balance. All right, so how do, what is the rate at how do all these processes work? Remember that the chemical buffer system is our first line of defense, only because it happens very quickly. Those chemical buffer systems happen almost immediately. Remember that double arrow, depending on what the body needs, is going to happen very quickly. We can convert those strong acids or strong bases to weak acids or weak bases. We can do that pretty much immediately. Those physiological buffer systems, those respiratory and renal mechanisms are going to happen a little bit more slowly. The respiratory component, I can alter my breathing rate and depth rather quickly. I can hold my breath and I can adjust my pH, or I can hyperventilate to adjust my pH. I can adjust my pH from a respiratory standpoint rather quickly. But from a renal perspective, from a kidney perspective, it's going to take longer for that to happen. I can't make my kidneys work more quickly. My body's going to have to take time to recognize that something's wrong for it to work. Now, and again, I'm not going to necessarily know that my body's in an altered state of acidity or alkalinity. So I'm not necessarily going to know that I'm going to have to adjust my rate of breathing or my depth of breathing. It's going to happen automatically. So that's why it says it's going to take minutes. If something's wrong, my body's going to try and do what it can to counteract whatever's going on inside my body. So from a respiratory standpoint, my body can say, hey, go ahead and alter your breathing. And it can do that in a matter of minutes, but it's going to take some time for your kidneys to kick in and do what it needs to do. All right, so we have, remember that the pH of our blood is 7.35 to 7.45, and we have conditions where the blood falls outside of those normal values. And if we are too acidic, we call that acidosis or acidemia, and that's when our pH falls below 7.35. So if we're below 7.35, that just means we have too much acid in our blood, or too much hydrogen ions, or too much hydrogen concentration of hydrogen ions in our body fluid. And it's either because we have too much acid or we've lost too much base. On the flip side, if we have gone above 7.45, we are in a condition called alkalemia or alkalosis. And that's because we've either lost too much acid or we've gained too much base. So we've decreased our hydrogen ion concentration in our body fluids. And we can break those up into a respiratory problem or a metabolic problem. So we can say we have respiratory acidosis, metabolic acidosis, respiratory alkalosis, or metabolic alkalosis. Now, I'll save that for when we get to that slide. All right, so here's just a kind of picture graph of, here's our normal 7.35 to 7.45. And if we are below 7.35, we are acidosis. And if we're above 7.45, we are alkalosis. And typically, we can't survive below 6.8, and we can't survive above 8.0. That's about our survival range, 6.8 to 8.0. For our blood pH. All right, and this is just a more, in-depth graph of kind of what we've just said, 7.35 to 7.45, and our average would be 7.4. So with acidosis, that's either because we have too much acid or we've lost base. But in either case, it's because we have too much hydrogen ion. And for alkalosis, we have a decreased hydrogen ion concentration because we've lost acid or we have too much base. Now, I said that we have two different types of acidosis, metabolic acidosis and respiratory acidosis. Now, I will say, so if you think of like where your belt is, your belt is at your waistline. If you vomit, the saying is, you lose acid above your belt, you lose base below your belt. So if you vomit, you're going to lose gastric acid. So if you lose acid, if you vomit, you're going to lose acid above your belt. So if you lose acid, you're going to become alkalotic, or you're going to go into a condition called alkalosis. You lose acid above your belt. If you lose base below the belt, so if you have diarrhea, you lose base below the belt. If you lose base, you're going to become acidotic. You've lost base. Going back to this slide, if you lose base, you're going to become acidotic. All right, so back to the slide. So respiratory acidosis. So respiratory acidosis is caused by a respiratory condition. That's the name. Respiratory acidosis is a condition that is that causes you to have a low pH, less than 7.35. That's caused by a respiratory condition. So acidosis is a lower pH. So that means we know we have a higher hydrogen ion concentration, which means we have a high CO2 concentration. And if we go back to our equation that we learned, we also have a high carbonic acid concentration. So respiratory acidosis caused by an increased carbon dioxide and carbonic acid levels increased in our blood. May include respiratory insufficiency, labored breathing, cyanosis, injury to respiratory centers and brainstem, obstruction of airway passages, disease that hinder gas exchange. All of these are respiratory conditions. That's the name respiratory acidosis. Metabolic acidosis are metabolic conditions, non-respiratory conditions. So accumulation of acids or losses of bases that do not fall under respiratory conditions. Kidney disease, where you can't excrete acids. Prolonged diarrhea. Remember, you lose base below the belt. If you've lost base, you have gained acid. You're keeping acids. Diabetes mellitus. Remember, if you've heard of diabetic ketoacidosis, DKA, diabetic ketoacidosis, in which acidic ketones are produced. That's a metabolic condition. So some of the symptoms of acidosis, you're going to see central nervous system conditions, drowsiness, disorientation, stupor, cyanosis. So when we talk about how we treat or how your body handles these conditions, if you have a respiratory condition, your body is going to handle that. It's called compensation. If your body is in a respiratory condition, your body is going to compensate metabolically. So we're going to go back up to some of these mechanisms. Metabolic, renal excretion. Okay. Respiratory, oh, nope. Since it's a respiratory condition, we can't treat a respiratory condition with a respiratory treatment. We can treat a respiratory condition with a metabolic process. A protein buffer system would be a metabolic process. A phosphate buffer system would be a metabolic process. These buffer systems are metabolic processes. If we have metabolic acidosis, if we have a metabolic condition, we're going to compensate respiratory wise. So if we have a metabolic condition, we are going to treat that condition by adjusting our breathing, our rate and depth of breathing. So if we have a respiratory condition, we're going to treat that metabolically. If we have a metabolic condition, we're going to treat that respiratory wise. All right. So here, some factors that are leading to respiratory and metabolic acidosis. So here you can see that if we decrease our rate of breathing, let's say that we are holding on to our breath, or we have obstruction of our airway passages, let's say we're choking, If we have pneumonia, we have decreased gas exchange. All of that's going to lead to accumulation of carbon dioxide. That's going to cause us to go into respiratory acidosis. So with all of these conditions, we're unable to blow off our carbon dioxide. So here we have an increased carbon dioxide level, which means we have an increased hydrogen ion concentration, which means we have a decrease in our pH. So decreased pH is our acidosis portion. This is a respiratory condition. This is a respiratory condition. This is a respiratory condition. So that's why we say respiratory acidosis. All right, kidney failure. Kidney failure, that's a metabolic condition. Failure to excrete acids. So we're holding on to acids. Excessive production of acidic ketones in diabetes mellitus. So again, we're talking about a metabolic condition. So we're producing acid. So non-respiratory acids. So now we're metabolic acidosis. Down at the bottom, prolonged diarrhea. So we've lost base. If we're losing base, it's a metabolic condition. So we're still metabolic. Prolonged vomiting. So we're losing acid. Excessive loss. So we're, I don't like this one. We're not going to worry about this one. Let's skip this example. Don't even worry about that example. All right, so we're gonna focus on this prolonged diarrhea. It's a metabolic condition. Remember, we're losing base. When we lose base, we're falling under metabolic acidosis. And remember, when we treat these conditions, we treat it with the opposite system. So if we have a respiratory condition, we're going to treat this metabolically. If we have a metabolic condition, we're going to treat a metabolic condition with our respiratory system. So with our respiratory condition, we're going to treat this with our buffer system and our kidneys excreting whatever it needs to secrete or hold on to. With our metabolic acidosis, with our acidosis, we might breathe off something, adjust our rate and depth of breathing. Respiratory infections can lead to accumulation of fluid in the alveoli that lead to decreased gas exchange. So I said pneumonia up here. It could just be another respiratory infection. All right, alkalosis. Remember, so alkalosis, respiratory alkalosis, excessive loss of carbon dioxide and carbonic acid. Hyperventilation. If you hyperventilate, you're blowing off a lot of carbon dioxide. It could be anxiety. Fever will cause you to breathe faster, poisoning, high altitude breathing. I don't know why playing a musical instrument is on there, but it was. Metabolic alkalosis, excessive loss of hydrogen ions or accumulation of bases, gastric drainage, vomiting, of stomach contents. Remember I said lose acid above the belt. If you lose acid, you're going to become more basic. Taking diuretics. You pee out a lot more. Taking excessive antacids. Remember, antacids are a buffer. Antacids will neutralize your stomach acid. When you neutralize your stomach acid, you are becoming more basic. And we're not just talking about, 1 or 2 Tums. We're talking when you take 9 Tums at once. And you do this, 3 or 4 times a day. If you have an ulcer and all you do is you just pop Tums and pop Tums and pop Tums, over time, you're going to come into a condition called metabolic alkalosis. And this is kind of what happens. This is the chemical equation of what you'll see when you take too many antacids. So some of the signs and symptoms of metabolic alkalosis, or just alkalosis in general: agitation, dizziness, lightheadedness, tingling. So with antacids, you're going to remember our bicarb equation that we learned in the respiratory system and here as well. But the bicarbonate ion binds to the hydrogen ion in the stomach fluid, neutralizing the acid, and it's going to raise the pH. And this is metabolic alkalosis. And remember, when we compensate, we're going to compensate a metabolic condition through the respiratory system. So we're going to, because we're in metabolic alkalosis, our pH is too high. So if we have metabolic alkalosis, we have a high pH, which means we have a low hydrogen ion concentration, and we have a low CO2. So what do we need to do if we take too many Tums over a long-standing time? We're in metabolic alkalosis, and we're going to compensate with our respiratory system. So we're going to decrease our breathing rate. So we're going to hold on to CO2. So CO2 is going to accumulate. Our hydrogen ion is going to accumulate. And hopefully our pH is going to, over time, decrease. All right. So here with anxiety, we're not going to go over all of these, but I think anxiety is the one that we can relate to the most. With anxiety, you're going to breathe faster. So think of hyperventilation. If you hyperventilate, you're going to blow off a lot of CO2. If you get rid of CO2, remember, decreased CO2 leads to decreased hydrogen ion concentration, which is going to increase your pH. So increased pH means alkalosis. And because we're talking about a breathing problem, this is a respiratory condition, respiratory alkalosis. On the flip side, vomiting. Vomiting, we've lost acid. If we lose acid, remember this is a metabolic condition. So we have a metabolic condition, we've lost acid. So if we lose acid, we've if we've lost acid, we have decreased our hydrogen ion concentration, so we have increased our pH. If we've lost acid, we've increased our pH, which is alkalosis. And because this is not a respiratory condition, it's a metabolic condition. So we're metabolic alkalosis. And remember, we'll talk about this one down here, administration of antacids. And again, we're not talking about 1 or 2 Tums here. We're talking about 9 Tums 3 to 4 times a day. So we're neutralizing our stomach acid. So when we neutralize our stomach acid, what we're doing is we're increasing our base. So if we increase our base, we are increasing our alkalosis state, and it's a metabolic condition. And when we treat a metabolic condition, we're going to compensate on the respiratory side. When we have a respiratory condition, we're going to compensate on the metabolic side. All right, so for compensation, remember it's going to be accomplished by changes in the action of various chemical buffers, the respiratory carbon dioxide excretion, and the renal hydrogen ion excretion. So we have the 3 things that we learned. We have these 3 things that we learned: the chemical buffer system, the respiratory, and the renal. So we know that if we have a metabolic condition, we know that the respiratory system is going to kick in. We know that if we have a respiratory condition, we know that the metabolic system is going to kick in. And then the chemical buffer system will actually kick in for both. And that will be all right, so with respiratory there we go. So with respiratory acidosis, if we are acidosis, we have too much acid. So we have too much hydrogen ions. The kidneys are going to get rid of hydrogen ions, so we're going to excrete those in the urine. The kidneys are not going to secrete the bases in the urine. The kidneys are going to hold on to the bases to kind of help neutralize those acids. And then the chemical buffer system is going to bind those hydrogen ions to help remove them from the fluids. And remember, we have these are our buffer systems: our bicarbonate, phosphate, and our protein amino acids. So with respiratory alkalosis, we have our decreased hydrogen ions. So our kidneys are going to retain hydrogen ions. Our kidneys are going to secrete bases. And then we have our chemical buffer system. And then with metabolic conditions, remember, those are going to be compensated by our respiratory system. And what's going to happen is if we're in acidosis, metabolic acidosis is we have too much hydrogen what we're going to do is we're just going to blow it off. Because remember, our hydrogen and our carbon dioxide go hand in hand. If we're in alkalosis, our carbon dioxide and hydrogen go hand in hand, so we're going to decrease our rate of breathing. And that's how we compensate. All right, so that is the end of this chapter. If you have any questions, please let me know, and I will see you guys in the next chapter. All right, take care.